Folks, and welcome everyone to another video lecture series demonstration from this particular channel, Bio-ProTech. So this is Pradyut Banerjee, your host. So in today's session, we'll be looking at two tools basically. Okay, so those are basic tools only, but are very useful. Okay, so when it comes to bioinformatics knowledge, when it comes to your gene-based analysis, okay, so today we are talking about, from the thumbnail you might have got already, but we are talking about your Gene Expression Omnibus. That is the first tool that we will be looking at, GEO. It is part of NCBI. And then we will also look at GEO, that is Gene Ontology. Okay, so the purpose of both of them are different. Okay, so GEO is mostly used for your gene expression quantification. Okay, so and your all of those things like gene related things like gene regulation, and you want to know about what is the analysis part of the NGS thing that has been done, or like from the NGS analysis what was been deciphered, or if you want to plot something. Okay, so if you want to plot something with that particular gene based data, that also you can do using GEO. Apart from that, if you want the raw NGS data that you are not gonna get in other places, so you're gonna get in GEO. Okay, so because you will be having the tar files and the zip files. Okay, so which are from the raw outputs from the NGS experiments. Okay, so if you want to practice them, okay, so this is the place to go. That is your Gene Expression Omnibus. You can go directly via typing GEO in your server, or you can go via NCBI as well. Okay, so I will show you both the ways. Okay, and after that we'll be looking at Gene Ontology. Okay, so what is Gene Ontology used for? So while we'll be demonstrating, I will let you know. Okay, so first things first, we can go to NCBI. Okay, so we can go to NCBI, and post that we can search by the name of any of the entries. Okay, so we have to select out of all of the databases, we need to select GEO over here. Okay, so here you can see GEO datasets and GEO profiles written over here. Okay, so we'll be focusing upon that. Either you go by the GEO datasets or profiles. Mostly we'll be concerned with the GEO datasets. Okay, so if you have the accession ID of it, so you can type that particular accession ID which starts with your GSE mostly, or you can directly type in the name of the organism that you want to search by. Like for this particular example, we can take the Mycobacterium tuberculosis. Okay, which causes tuberculosis. Okay, so I have the session ID by my side for this particular demonstration. So I would go by that. So or you search via the Mycobacterium tuberculosis, you will be getting multiple entries under that. You can select any one of them and go inside that and see that what is the paper published, what are the things that you can get from there, what sort of information you are getting. Okay, but our main aim over here is to do something using a tool called as GEO to R. GEO is the dataset name. Okay, and GEO to R, basically that is a R interface, R language we are talking about. So if you don't know how to write in R, or how to write your codes in R, so this particular tool can help you with that. Okay, because for this you don't need any of the coding knowledge. Okay, so it is running in the background. You just have to select the control test this that. Okay, and then it will give you the gene expression quantification, as well as the figurative representation or the graphical representation of the same. Okay, so that you want to put anywhere, all of the p values, everything it will give you. Okay, so I will show you the same over here. Okay, so this is the ID. If I search via this, okay, so this is one way. Another way is, of course, by directly typing in the GEO. Okay, so in the Google, and you will be redirected to the GEO page, and there also you can search. Okay, so yeah, but it is hosted by NCBI anyways. So doesn't matter like via which particular means you go, you'll land up to the same sort of results. Okay, so you can basically go to GEO home, because here they are showing one by one, so we don't intend to do so. Okay, so here you can go to GEO home, Gene Expression Omnibus. Here you can see, here you can type the same thing. Okay, so GSE, Okay, 216603. Yeah. So once we do that, here is the entry that we were talking about. Okay, so what sort of information you're getting? So let's have a glimpse of that first. Okay, so this is the NCBI site, as you can see. So this is the paper that is published. The organism name is Mycobacterium tuberculosis. I have already told you that. Okay, and this is the summary. So what is this particular summary all about? This is just like an abstract, but this is not an abstract. This is the summary of the things which are needed for this particular exercise, or basically to know about the gene expression quantification and expression analysis. Like what is the summary of the what do you say, the experiment they did. Okay, like how many controls they took, how many experimental setup they have done, and what were the results. So that is the gist. In the gist format they have provided this particular thing. You can read like all of these. Okay, so that will help you a lot. Apart from that, you can also go to this particular PubMed link. Of course, the full link to this particular paper is also given. You can also consider going to that and reading the whole paper if you want to. Okay, but the main thing that we are concerned about over here, if I go to the samples, the same thing is written over here, but the samples that they have come up with are being enlisted over here. Okay, as well as the platform information is also given, via which particular platform they were able to do this particular NGS, and likewise all the submission, like the in pointers it is being given for your ease, like for your ease of understanding. Okay, here you can see the country US, and then like contact name, email ID of that particular author, or like first author or corresponding author is being given. So likewise submission latest date. So we are not concerned about this particular part though. Okay, so we are mostly concerned about this particular part. Okay, so these 6 things that you see over here are of our importance. Okay, so what is this particular thing was single, or you are considering all of them? What are these things? Okay, what are they used for? Basically, if you read this particular paper, you will get to know that out of this, some are control, some are test. Okay, so based on that, we'll be doing this particular analysis. Okay, and in order to do so, we can go via this particular tool, analyze with GEO to R. This is what I was telling you like at the start of the video. Okay, so here you can see the particular raw NGS data that you get from the experiment, like like NGS is being used, or the 3rd gen sequencing is being used, or the 1st gen sequencing even is being used. Any kind of sequencing platform, okay, is being used, and you are getting the what do you say, the raw output of that particular thing over here. Okay, so you can consider downloading this particular thing in tar file, or why this is of so much of a small size, because it is a zip file. Here you can see .tar file is written over here already. Okay, so it is a compressed file. So compressed format is there. So but if you download it and if you double click on it, so you will get the whole file by your side, and using that you can analyze that particular data using Galaxy server, also using R or Python, also the NGS data analysis, the regular NGS data analysis pipeline that you follow, like initially doing the like a quality control, then you're trimming the trimming the adapters, and then you do all of the stuff like alignment and this that. Okay, so then you land up to the final step, that is your gene expression quantification and this that. Okay, so here you don't have to do anything. All of the things are automated, like backed by R. Okay, so R will be doing the stuff for us. Just we have to define the groups. Okay, so we will hit on analyze with GEO to R. One more note is that in all of the data sets, you will not be finding the GEO to R option, by the way. Okay, why is it so? Because the clear differentiator of the control and the control and test are not provided over there, because of which the GEO to R cannot be utilized to analyze the same. Okay, but many of them will be finding this particular tool. So we are talking about this particular tool only. So we'll go by this. Otherwise, you can always do the analysis, like taking that particular raw data, downloading it from the bottom that I have shown you. Okay, so you can take that particular data and analyze it using any of the pipeline. Okay, or use the standard pipelines from your like using the tools like Galaxy Server, or your like R or Python. Also you can utilize the language you can use for your data analysis. Okay, so now we have to define the groups. So I would define the groups as, let's suppose, control. Okay, one group name is control, another group name is test. Let's suppose. Okay, so here you can see the color connotation by that. So like control would be of this color, test would be of this color, so it would be a clear differentiator. Okay, so if we see this one, okay, so this one is experiment. Let's this one control. You can read that which one are the control, which one are the test. Okay, so I'm just doing it for the example sake to make you understand. So these three I have labeled as control, whereas the other ones are my test. It can be other way around also. I'm just like doing it for the example sake. Okay, so after this you can directly hit on analyze. So it will analyze via the default settings that it has, which is good enough. Okay, otherwise if you want to change the like how the p value is being calculated, which particular type of method that you want to employ to calculate p value, so that you can change from here. Okay, and all of the things that you can customize to. Okay, for genomics you can also change the significance levels from 0.05 to 0.03, because in genomics mostly we follow 0.03 instead of 0.05. Okay, but by default, let's see by the default what it gives us. Okay, so yeah, so we can go by that. Okay, and you can see also see the R script that is going in the background. Okay, so this is the R script that is going in the background. You need not to code anything. Okay, so yeah, of course we will come up with much more session on this particular coding part also, Python, R, everything in this particular channel, okay, in the future. So as of now I'm just stating for the beginners who doesn't have any background of coding, so they can also utilize this particular tool and do their analysis. Okay, so if I hit on analyze, okay, so I haven't changed the significance value. It is still that 0.05 only. Here you can see. Now here you see the top differentially expressed gene. So what is the main purpose of ours when we do a data analysis, like any kind of sequencing data analysis? The main thing that we want to look uh, at is the differentially expressed genes, and these are the top differentially expressed genes. Okay, so this is the representation in a tabular form, and at the top you see the same representation in the form of graphs. Okay, so this is a volcano plot. Okay, so this is a mean difference plot. Okay, likewise you have U map is there. Okay, so then you have your different sort of plots are there. Venn diagram is there. Okay, then you have box plot, then your density plot, p value histogram, jitter plot. Okay, likewise you will be having. Okay, so this is a mean variance trend plot. Likewise, one by one also you can go, like you go by the IDs of it. You can see what are the positively expressed and negatively expressed genes. Okay, so likewise here you can see. Okay, so expression values here you can see. I think we have selected the other way around, so that is not a problem. It is just for the example sake I'm giving you. Okay, so it would be other way around. This would be a test, this would be a like control, and likewise. Okay, so that for that you need to like read that particular paper. For each data the thing would be different. Okay, so that is why one by one also you can spot the sample. Okay, so sample values here you can see like the positive here in this particular expression. Here you can see in the controls all of them are positively regulated, whereas the in the test negatively regulated. Here you can see. Okay, so what are the possibilities? Gene title, by that also you can decipher what is going on. Okay, so samples that you have defined, if you want to see again. Okay, so redefine group that you want to do, that is also possible if you want to redefine it. Okay, so likewise, so basically in this particular way you can see how to do this particular expression quantification or differentially expressed gene you want to look at. You can see the adjusted p values. Okay, so t like log fc values, all of these are provided over here. Okay, so p values everyone must be knowing, so that the significance value that we look at. Okay, so p value is the probability value. Basically we compare it to the significance value, and if it is like greater than that particular thing, so we like 0.05 and all, so we consider the, like we have enough evidence to reject the null hypothesis, okay, and accept the alternate hypothesis, okay, as we know. Because what does the null hypothesis says? Null hypothesis says that there is nothing happening, okay, null, look, okay, so there is no change, so whatever you are doing has no effect, okay, or has a, like has no effect basically, okay, so neither positive nor negative, so that we don't want as a researcher, no, so we want some effect, either it be a positive or a negative, we want some effect. So we want to accept the null hypothesis, and we should be having the enough evidence to accept the alternate hypothesis, sorry, yeah, so we want to accept the alternate hypothesis, okay. And what does the alternate hypothesis says? The alternate hypothesis says there is some effect, either it is positive or negative, it is not saying, but it is having some effect, and for that we need to have enough evidence to reject the null hypothesis, okay. And for that we need to have the like significance values, like the p value greater than significance value, and of course that is more than 0.05 and likewise, okay. So yeah, so that is the part that you can do using GEO and GEO to R. So GEO is the place to get your raw sequencing data from. GEO to R is the tool to analyze that particular thing for looking for your top differentially expressed genes, and the figurative representation you want to do, or basically graphical representation you want to do, or the tabular form you want to know the p values, everything it will help you with, okay. Without any knowledge of R, it like you are utilizing R, but without any knowledge of R also you can pursue the same, okay. So that is about the GEO, okay. Now let's go on to the next tool that we were talking about, that is your Gene Ontology, okay. So GO, if you type, so it might redirect you to some other page, so better write Gene Ontology, the whole thing, okay. So Gene Ontology resource, okay, so if you go inside this, okay, so you would be finding that there are two search two search engines that you can find over here, okay. So two search option that you are finding. If you want to know more about it, you can always go to about what is GEO, okay. So you should ideally go and read what is the mission of this particular GEO, what is the meaning of Gene Ontology, okay. So this is the main thing that you have to read, okay. So what is gene annotation, okay, so and what is Gene Ontology? Basically Gene Ontology is a logical structure describing thing, okay. So which basically categorizes any of the biological thing into three categories majorly. One is called as your molecular function, second is your biological process, third is your cellular component. Mostly this particular Gene Ontology is used to know the relevance or the functioning of a gene at the metabolomic level. Mostly we use it at the metabolomic level, but not necessarily though, okay, because we can know that that particular gene is part of which is taking part in which particular molecular function, which particular biological processes it is involved in, okay. The cyclic process, basically the metabolic processes we are talking about, okay. And like which particular cellular component it belongs to, okay. So that is the main thing that we want to look at. So it is like published in Nature Genetics, okay. So you can read that particular paper that was published, okay, way back, okay. So you can read that particular paper also if you want to. Apart from that, if you want to know the Gene Ontology term or the overview of that, you can go via this, okay. So what is the meaning of your molecular function, cellular component, biological process, how it is categorizing, what is the root, what is the hierarchy, how it starts, what is the root point, and from where it starts, okay. So all about those you can read, okay. So likewise, now let's do the thing that we want to do, basically search, okay. So here you can see current release, so of course it keeps on changing as per the new research is being done, so new entries are being done, so based on that this particular number tends to of course change, okay. So you can see more than 16 lakh gene products are there, okay. So 92 lakh 81,704 annotations are there. Annotation is nothing but a unique identifier or unique ID that has been given to them, so that we can, if we search via that particular ID, so we'll land up to that particular thing only. Okay, so more like almost 5,500 species are being listed over here. Okay, then 39,354 geo terms are being defined over here. Okay, but it will categorize again in the three sects, that is biological process, molecular function, and cellular component. So that is the main thing. Okay, so this is geo enrichment analysis powered by Panther. So this is the Panther server that would be utilized, okay, to like give you the results, whereas in this one you can directly go by the ontology terms. Okay, so we will search via both and see what are the differences or the like commonalities that we are going to get. Okay, so we'll come up with our own gene we can come up with. So let's start the example with BRCA1, BRCA1, okay, which causes breast cancer. Okay, okay, some rate limitation, I think so. There is some issue with the server. Nevertheless, okay, we'll try once again. Okay, so security issue is there for this one, so that won't be a problem because I have already clicked on that. So this time around it will show. Yeah, so here you see this is the thing, but for the PNPLA3 they don't have any results. Here you can see they don't have any results for the same. Okay, so let's look at the some different try. Okay, so let's try something different, like PNPLA3 we have tried already. GATA3. Okay, GATA3 results are also not there. So likewise, we need to see, because it's the ontology terms. Okay, so based on the ontology terms, we have to search. Or if you go by any, so there it will give some results because it is gene or gene product, and based on that it is giving. Okay, so let's search by any as well. Okay. So here you can see, because in the ontology terms it was having 0 entries, so that is why not giving us answers. But in the annotation it has almost near about 2000 entries, whereas the gene and gene product it is having 27 entries. Okay, so we can also go by the gene and gene product, because what it codes for, or what it codes for, or basically what it like influences, that is also very important. Or if it like, of course it codes for a protein. Okay, so that particular protein might be of our use. Okay, so you can go by the source of it. Okay, then the organism name. If you want to don't want to include or basically filter out certain things, so like mammalians you don't want to include, you can do a minus, so the mammalian result would be gone from here. Okay, here you can see only gallus and this that are there. Okay, so like the mammals result would be gone. So likewise you can like apply the filters. Okay, so here you have in the Panther family classification you have two type of filters. Okay, so what is this particular classification? Panther family classification is a type of classification. Okay, so in protein also we have multiple different ways of classifying things, like PFAM is a technique, like P family, basically that is a class what do you say, a technique for your family based classification. Okay, so you have your GATA transcription factor is there, then your transcription factor, GATA family membrane protein. Okay, so we have, so based on that you're having this particular search result. If you want to like not include any of those, so you can obviously click on or deselect them basically. Okay, so from here. Okay, so these are the things that you can include, or what do you say, you can do a minus from that. Okay, so here you can see unused filters. Okay, so we haven't used this particular filter. Apart from that, you can go inside this also, looking at like what is this entry all about and what are the things it is associated with. Okay, then the like geo class, what are the classes that it comes under, like the regulation, what is it doing. So how many biological processes it is involved in, here you can see 177. How many cellular processes it is involved in, 110. Okay, and likewise you can go inside that particular thing also. Okay, so if you go by this, okay, so here you have the reference IDs. Okay, so via that here only, like, see, the total annotations are 212, so don't get like fooled by the this particular 10 entries only. So you can select here and you can go to 100 also. Okay, so if you go to 100, it will show you 100 entries at a time. Then you can go to the next page likewise. Okay, so then you can do next. Previous next is there na? So next you do, so then 101, from 101 it will start. Okay, so you can see from 101 to 200, there is the next thing. Okay, so what is the broader panther family classification that you can see from here? Okay, which particular organism it belongs to, then the geo class that it is a part of. Okay, all of the things that you can see from this one. Okay, so that is there. Okay, so now if we go back, okay, so likewise you can go. Okay, so annotation, if you want to go, what are the gene and gene product annotation that you're getting? Okay, so those are the things that you can do using gene ontology term. Okay, so if you search via some other gene also, you will land up with the ontology terms as well, that what it categorizes into. Okay, so likewise, okay, now let's do the same thing, use the same gene and search in here. Okay, so we also have some examples, though we can go by the example first. Okay, we can go by the example and launch, instead of biological process, let's do a molecular function. Okay, from the homo sapiens only, although you can choose from different species. Okay, so from here, okay, so we stick to homo sapien and we do a molecular function, and the exemplary genes we are taking a set of genes, basically a pool of genes. So you can just have to click I am not a robot and then continue with the same. Okay, so here you have the entries. Okay, so here you see the classes and subclasses over here. Okay, so geo molecular like function complete terms that you are getting over here. Okay, so which particular functions they are involved in, so that is why this particular thing is most widely used in your metabolomic analysis, that it is like involved in which particular processes. Okay, so the fold enrichment is also given over here. Okay, so like you can see here you have the platelet derived, your growth factor binding, then growth factor binding 136, and like reference values you are finding. Okay, signaling receptor binding, you have more than 1500 entries. You can go inside that also and you can see what are those entries all about. Okay, the families, everything you can see. Okay, so protein binding, what kind of things that you get inside this. Okay, so it depends upon your research question what you want to find. Okay, so I can just show you the way. Okay, but what is your research question, based on that you would be searching now. So based on that you will land up to that particular result. That, let's suppose you want to know that molecular function regulatory activity, so this many are there. Okay, then like receptor ligand activity, let's suppose we are doing a drug discovery, so we want to know about this. Okay, so we go inside this, we have near about 559, not near about, exactly 559 entries over here, so mapped genes that are being over here. Okay, so based on that you can see, okay, you can see all of those results. So you can use a representation that you can easily use for representing all of these. Instead of tabular form, it would be easier to see them in a like a like pie chart or a bar chart. Okay, so this is a like a pie chart that you see. Unclassified we don't want anyways, so unclassified, that is the major chunk. We want to remove this. Okay, so filter unclassified, because unclassified means nothing anyways. Okay, so the total functional hits now you are getting, so from the 559 genes you are getting a functional hit of 583, because one gene might be involved in multiple processes as well, that is why. Okay, so here you can see what are the subcategories of processes that it is involved in. Based on that you have the pie plot plotted over here. Apart from that, if you want to go on to the cellular component, that also you can go. Okay, here you can see most of them are involved in your cellular like anatomical entry and your protein containing complexes. Okay, so which is of like a smaller number. Okay, so molecular function we have already looked at, I reckon. Yeah, So we'll look at biological processes. Okay, so these many biological processes it is involved in. Okay, so unclassified we have already filtered out. This is now you can see biological process involved in which particular thing: cellular process, developmental process, homeostasis process. Okay, so and that particular color concerns to like how much of a percentage it is in there. Okay, so here you can see much more hits we are getting from the same number of genes. Of course the number of genes are not going to change, and we are getting so many like total process hits. Why? Oh, because I have already told, a single gene might be involved in 2 or 3 processes or more than that also. Okay, that is why, or they are part of multiple categories. One gene can be a part of all multiple categories involved in multiple of the functioning or multiple of the biological processes. That is why. Okay, similarly you can do the last but not the least, I will show you using this custom example instead. Okay, so custom example we can go by. Let's do that. Instead of using their example, we can come up with the same examples that we were the genes that we were talking about. We'll take some random genes which doesn't have any correlation with themselves, like BRCA1, BRCA2 involved in your breast cancer, PLA3 involved in your non-alcoholic fatty liver disease, GATA3 involved in your stomach cancer and all. Okay, so we will like to see that what are the biological processes they are involved in. It is not that all of them would be involved in the same biological process. There might be two of the genes involved in a single process, or the all of them might also be. So we need to see that. Okay, so that is why it is a very essential part of metabolomic analysis that we do. Okay, so we are using the Panther classification system. So why this particular classification system? Because of course any of the thing which has a family starts with a root node, and likewise it goes beyond. Okay, so then you have the categories and the subcategories. Okay, so here you can see, as we have used BRCA1, and the BRCA1 abnormality or basically anomaly in that particular gene leads to breast cancer. Okay, so here you can see 5 of them are involved in negative regulation of mammary gland. Why? Mammary gland is part of your like breast, it like when you have the breast cancer, you will have the negative regulation of mammary gland, so that like the what do you say, the milk secretion or this that would be hindered, and this that. Okay, likewise you will be having the lump and all. Okay, regulation of mammary gland epithelial cell proliferation. Okay, so cell proliferation would also be aggravated because it is a cancer, as you understand. Okay, so they are involved in such things. So that is why here you can see then the regulation of DNA damage checkpoint. Of course, if the DNA damage checkpoints were right, so then we won't have got the cancer mostly. Okay, so that is why there also certain abnormalities are expected to happen. Okay, likewise then respond to ionized radiation, because all of this are mostly involved in cancer. That is why here you can see all of the negative regulation are being pointed out over here. Let's have a look at the regulation of mammary gland epithelial cell proliferation. Okay, 16 outputs that we are having. Okay, so out of which, so this is the what is a tabular representation of the same, which particular part, like which particular gene ID is involved in which particular process. Okay, some of them are here you can see are like part of, like, of course, so BRCA1 and BRCA2 would be coding for their own gene, like this is the BRCA2 gene susceptibility protein. Okay, so not gene coding for the protein. So BRCA2 protein, likewise you also would be having the BRCA1 protein. Okay, so like here you can see RASP responsive element binding protein 1, although you should also be having BRCA1 protein, but it is not seen over here. Might be that it is not involved in the second process, it is involved in the first process, that is the negative regulation of the breast cancer. Okay, so apoptosis regulatory backs it is involved in. Okay, so likewise, so you can do a pie chart representation of the same. Okay, so the same 16 entries would be shown, 16 genes, but here you have the hits as 33, because the same reason multiple processes a particular gene can be involved in. Okay, filter unclassified. Okay, so that we only get the results that we intend to. Okay, biological regulation, cellular process, developmental process, immune system It is involved in the green, represents that. Okay, response to a particular stimuli, that is your represented in your pink. Okay, so if you point your cursor towards here also you can see the GEO ID, annotation ID. Apart from that, what is the percentage in the whole 100%? So it is 18.8%. Okay, so here you can see likewise 62.5% over here. So likewise this is 9.7%. Okay, so what is this? Without unclassified and with classified, so it is showing. Okay, so that is why. Now you can change it, of course, the protein class. What are the protein classes that you have? Okay, so here you can see these are the protein classes inside this. Like then pathways it is involved in, that is also very important at times. Okay, so you can go to a like bar chart instead. Okay, so if you want to do so, so you can increase the size of it to 150%, so it will basically increase the size. Okay, or zoom it out, nothing fancy. Okay, so here you can see like these are the pathways it is involved in. Okay, so this is very much useful when it like when you want to count upon the pathways it is involved in, and you are doing a metabolomic analysis which genes are involved in, what kind of a pathway. Although for that you have a number of different tools also, okay, like the reactome to the keg to all of those. Okay, but like gene wise if you want to search, so GEO can help you with that. Okay, apart from that, of course, the main 3 GEO terms that are there, always keep in mind. Okay, that is your molecular function, biological process, and cellular component. It will always try to divide any of the, or basically segregate any of the data within this particular 3 components. Okay, so yeah, so that is all from my side. So this is Pradeep Banerjee signing off. See you in the next one with some quick demonstration. So this is one of the basic bioinformatics tool demonstration or web server demonstration we have done. Okay, serving 2 different purposes. Hope you have understood something new. If you are getting some knowledge out of this particular channel, do consider subscribing to the channel and hitting the like button. Okay, and sharing the videos with your peer ones. Okay, yeah, so that is all from my side. Okay, see you in the next one. Bye bye, take care.